Okay, should I start?
1 minute, ma'am. Yes, ma'am. Now you can start the class, ma'am.
Good evening. Welcome to our exam mastery live classes. So today we'll be talking about nucleic acid, their structures and nucleotide metabolism. So introducing you to this mastery live classes series. This series is based on rapid revision of all important topics. All volatile facts will be quickly covered. Then all high energy, high yield topics and all the topics which are frequently asked in the examinations. We're trying to cover that in this series so that it's going to benefit your university exams. I'm going to take this class in question and answer sessions, like long and short question and answers that are frequently asked. Then introducing you to the core features of Oracle. Here at Oracle, we have live classes to elevate your learning experience. Then apart from that, we also provide you with recorded lectures so that you can access those lectures as per your convenience whenever and wherever you want. And apart from that, we have also launched our notes, which are available now. So it is a very comprehensive book based on latest CBME syllabus. All bulleted out points and everything is given in the notes in a very precise form. It's very colorful also to keep you interested. Then apart from that, we also have MCQs so that you're able to assess the knowledge that you gained. Then we have three different subscription plans at Oracle, starting from the basic plan, which is for 3 months and comes at 15,000, followed by the standard plan, which is for 6 months and comes for 30,000, and then the mentorship plan, which is for 12 months and will cost you 50,000. So in the mentorship plan, we also provide you with personalized guidance as well as notes. Then you can get additional 20% off by using my promo code as displayed on the screen. So let's start with today's topic, that is nucleic acid structure and nucleotide metabolism. So your nucleic acids, they are polymers of nucleotide. Because if you have to give a one-line definition, you have to define nucleic acids. So what are they? They are polymers. Polymers of what? Polymers of nucleotides. Okay, therefore they are also known as polynucleotides. Poly means many and nucleotides because there are many nucleotides joined together. Just like if you remember when we studied glycogen, I said glycogen is a polymer of alpha D glucose. Okay, you are putting together many glucose molecules to form a polymer of glycogen. Okay, similarly, you are putting many nucleotides together to form nucleic acid. Is just like synonymous to putting amino acids together to form proteins. Your proteins are polymers of amino acids. So here the building block of nucleic acid is your nucleotides. Okay, so it's something like this. This is one nucleotide, another nucleotide, another nucleotide, another nucleotide, another, and you end up with nucleic acid. Okay, so these individual units, these monomeric units, they are known as nucleotides. Okay, so very simply, what are nucleic acids? They are polymers of nucleotides and they are of two types. Okay, now your nucleic acid, it can be of two types, that is RNA and DNA. RNA is your ribonucleic acid and DNA is your deoxyribonucleic acid. Acid and deoxyribonucleic acid. Okay. Now let's talk about what are nucleotides. Let's define this nucleotide. So moving forward, talking about nucleotides. So your nucleotides, as I said, these are the monomeric units of nucleic acid, okay? And they are composed of three components. What are they? So your nucleotide, it consists, number one, consists a nitrogenous base. Then it has a pentose sugar. Pentose sugar means a sugar with 5 carbons. Okay. So a 5 carbon sugar, that is a pentose sugar. And lastly, it has a phosphate. Okay, and iron or phosphoric acid. So these are the components of your nucleotide. At first, I said that your nucleic acid, it is made up of monomeric units, nucleotides. Now these nucleotides, they are made up of nitrogenous base, pentose sugar, and phosphate. So all these three things put together, they form your nucleotide. Okay, and a nitrogenous base and a pentose sugar, just the two of these, they form what is known as nucleoside. Okay, so nucleotides are phosphate esters of nucleoside because this is a nucleoside. Your nitrogenous base and pentose sugar, I'm saying that this unit is known as a nucleoside. And when to this nucleoside you add a phosphate, it becomes a nucleotide. So in other words, what else can you say? That your nucleotides, you can also say that your nucleotides are phosphate esters of nucleosides. I hope it's clear to you. And once when I define nucleotides and nucleosides, all the structures become more clear. Okay. So for now, just remember that you have nucleotides, and nucleotides, they are made of nitrogenous bases, pentose sugar, and phosphate group. These three components, they are making your nucleotides. And when multiple nucleotides they are put together, they form a polymer which is known as nucleic acid. And your nucleic acid is of two types, RNA and DNA. How do you define RNA and DNA? That is on the basis of the sugar, the different types of sugar. I'll also define the sugars later in the lecture. And your nitrogenous base, when it is just attached to the pentose sugar, it is forming a unit which is known as nucleoside. Now, to this nucleoside, when you add a phosphate to an ester bond, you end up with a nucleotide. So you can also define your nucleotides that they are phosphate esters of nucleoside. Okay, I think this is very simple. Now, moving on to the individual components of your nucleotide, taking them one by one. So beginning with the nitrogenous base. So what is a nitrogenous base? They are heterocyclic, planar, relatively water insoluble, aromatic molecules. Okay, so these are planar and heterocyclic structures, and these are aromatic molecules. Okay, now your nitrogenous bases, they can be of two types, pyrimidines and purines. Okay, so you both your DNA and RNA, they contain pyrimidines and purines. So your nitrogenous bases, they're of two types, that is purine bases and pyrimidine bases. Now let's talk about the purine bases. So it is a 9-membered ring, and it has two rings in its structure. Although you can remember this, that your purine, this is a short word, but it has two rings. Pyrimidine, it is a lengthy word, but it has one ring. Okay. So your purine bases, they are 9-membered rings, and it has two rings, which is composed of carbon and nitrogen. And the two common purine bases found in DNA and RNA are your adenine and your guanine. Your adenine is 6-amino purine. That is, to the purine ring at the sixth position, you have amino. And your guanine is 6-oxy 2-amino purine. Okay. And to remember that your purine bases, because usually students get confused whether cytosine is a purine, whether adenine is a purine, or it is a pyrimidine. So there's a mnemonic that I'm giving to you. All girls are pure. Now, Pu is for purine and A for adenine, G for guanine. So just remember this mnemonic, all girls are pure. So Pu is for purine. So your purine, it has adenine and guanine. Okay. So that is the mnemonic to remember. Now, if you have a look at the structure of purine, it will become more clear. So like I said that your purine, it is a 9-membered ring. So you can see 1, 2, 3, 4, 5, 6 members. Okay, so in the first ring, the cycling, the numbering is in this way. That is anti-clockwise numbering. And in the second ring, this you have 7, 8, and 9. So the numbering is like this. Okay, so in the second ring, the numbering is in the clockwise manner, clockwise numbering, and in the first ring, it is in the anti-clockwise manner. So the first ring, the numbering is anti-clockwise. Okay, so this is your purine ring, and as you can see, it has two rings in structure composed of oxygen, composed of carbon and nitrogen. So there's nitrogen at first position, as third position, as seventh, at ninth. Okay, and rest you have carbons. Okay, so this is your purine ring. Then we said that adenine and guanine are the two most common purine bases. Okay, so this is your adenine and this is your guanine. Now you can see in the adenine you have this amino group at 1, 2, 3, 4, 5, 6. So therefore it is 6 amino purine. Your adenine is, like I mentioned here, your adenine is 6 amino purine. Okay, and your guanine is 6 oxy 2 amino purine. Let's have a look. 6 oxy. So at the sixth position you have this carbonyl group attached, 6 oxy and 2 amino. So at the second position, you have amino group attached. So that is guanine for you. Okay, 6 oxy, 2 amino, guanine. I hope purine is clear to you. Then you have some other minor purines as well. So these were the two common purine bases, that is your adenine and guanine. Then you have some minor purines as well. What are they? Okay. So your minor purines, they are your xanthine, your hypoxanthine, and uric acid. Although uric acid is a purine degradation product, but the purine ring is intact in it. Okay, so we'll consider it as a minor purine. Okay, so your xanthine is 2,6 dioxopurine. That is at the second and the sixth position, you have two carbonyl groups attached, so it becomes xanthine 2,6 dioxopurine. Your hypoxanthine is 6 oxopurine. And your uric acid is 2,6,8 trioxopurine. Okay, so I hope now the structure of purine is clear to you. Let's have a look at it again. Here, it is heterocyclic. It has two rings. Okay, 9-membered. In the first ring, the numbering is anti-clockwise. In the second ring, the numbering is clockwise. Then you have two most common purine bases, that are your adenine and guanine. Okay, and you can remember it by the mnemonic that I've given. All girls are pure. So your purines, the common ones are adenine and guanine. Then you also have some minor purines like xanthine, hypoxanthine, and uric acid. Okay, so that was about purines. Now moving on to the next nitrogenous base, that is pyrimidine. So your pyrimidine is having a single heterogeneous ring. And how are you going to remember this? Pyrimidine is a lengthy word, but it has a single ring. Purine is a short word, but it has two rings. Okay. Now your common pyrimidines, these are your cytosine, your uracil, and your thymine. Okay, so your cytosine, it is present both in DNA and RNA. And your uracil, as we all know, it is only present in RNA. And your thymine is only present in DNA. Okay, now let's have a look at the structure of pyrimidine. So this is your pyrimidine ring. This is how it looks like, a single ring numbering 1, 2, 3, 4, 5, 6. Okay, so this is your pyrimidine ring. Now the common pyrimidines that I told you, they are your thymine, cytosine, and uracil. Okay, so your thymine, this is having, as you can see, a methyl at the fifth position. So it is also known as, the other name for thymine is 5-methyl-2,4-dioxypyrimidine, because at the fourth position and at the second position you can see C double bond O. So your thymine is 5-methyl 2,4-dioxypyrimidine. Now talking about cytosine, your cytosine is 2-oxy, as you can see at the second position, C double bond O, 2-oxy, 4-amino. So this is 1, 2, 3, 4, 5, 6, actually 6 amino. Okay, 2-oxy, 6-amino pyrimidine. That is your cytosine. And then uracil. In uracil and thymine, what is the only difference? There is no methyl here in uracil as there was in thymine, you can see. Okay, that is the only difference. Otherwise, there is this C double bond O at second carbon, and at fourth carbon, you can see there is C double bond O. Okay, so that is the only difference. Otherwise, your uracil and thymine, they are almost having similar structures. That is that your uracil does not have a methyl. So your uracil is 2,4-dioxypyrimidine. Okay, 2,4-dioxypyrimidine. Here, sorry, the numbering is from here. Okay, 1, 2, 3, 4, 5, and 6. This is the correct numbering. Okay, 1, 2, 3, 4, 5, and 6. Okay, so your cytosine, it is 2-oxy 4-amino pyrimidine. I'll write it 2-oxy, as you can see, 2-oxy 4-amino pyrimidine. Okay, so that was about the pyrimidines, thymine, cytosine, and uracil. So with this, we come to the end of nitrogenous bases. So we've taken the first component of nucleotide, that is nitrogenous base. Okay, now moving on to the pentose sugars. So your pentose sugars, these are 5-carbon sugars, therefore known as pentose. These are naturally occurring nucleic acids. Sugars, okay, so the naturally occurring sugars in your nucleic acid, there are two types, that is ribose and deoxyribose. So you have ribose sugars and deoxyribose. So your pentose sugars, there are two types, ribose and deoxyribose. And what is the difference between ribose and deoxyribose? That is in the presence of the hydroxyl. Okay, so the 2' hydroxyl in ribose. So in ribose, at the 2' position, there is a hydroxyl group, whereas in deoxyribose, that 2' hydroxyl, it has been deoxygenated and there is only hydrogen present. So in deoxyribose at the 2' position, only hydrogen is present, whereas in ribose sugar at the 2' position, there is hydroxyl group present. So your hydroxyl, it is deoxygenated at second position in deoxyribose. If I show you the structures, it will become more clear to you. See, this is the structure for ribose and deoxyribose sugar. So as you can see, in the ribose sugar, this is the numbering at the 2' position. This is carbon 1, carbon 2, carbon 3. Okay, so at these 2' position, you can see in ribose sugar, you have hydroxyl group. Whereas in deoxyribose at the 2' position, in that sugar, in deoxyribose sugar, the 2' hydroxyl has been deoxygenated and you only have hydrogen presence at 2' position. Okay, this is your 2' position. So that is the difference between ribose sugar and deoxyribose sugar. So your DNA, they are having deoxyribose sugar. So your deoxyribose sugar is present in DNA and your ribose sugar, it is present in RNA. Okay, so that was about the sugars. I hope now it's clear that your nucleic acid made up of polynucleotides, that is, they are polymers of nucleotides. So nucleotides are having nitrogenous base. I described the nitrogenous bases that there are two types, pyrimidines and purines. Okay, your pyrimidines, they are having two rings in its structure. It is heterocyclic. Okay, and the most common purine bases, they are your adenine and guanine. Then the pyrimidine bases, it is heterocyclic single ring structure. Okay, and the common pyrimidine bases, they are your thymine, cytosine, and uracil. Then moving forward to the types of sugars, there are two types of sugars, ribose and deoxyribose. What is the difference between the two sugars I've described? That is 2' position. In the ribose sugar, you have hydroxyl, whereas in deoxyribose, you just have hydrogen. Now moving forward to nucleosides. Okay. Now nucleosides, like I told you in the beginning, your nitrogenous base plus pentose sugar. This forms a nucleoside. So let's see how it's going to be formed. With the help of a diagram, it's going to be more clear. So this is your nitrogenous base. I hope you can recognize which nitrogenous base this is. This is 6-amino purine. So this is adenine. Okay. Now adenine, this is the nitrogen 9, because if you remember the numbering in the second ring, it was 1, 2, 3, 4, 5, 6. This was 7, 8, and 9. Okay, so this was your nitrogen 9, and this nitrogen 9 is connected to the C1 of the pentose sugar. You can see this is a pentose sugar and this is having hydroxyl. So this must be ribose sugar. Okay, now you can recognize that to the carbon 1 of this sugar, what is connected? The nitrogen 9 of your purine is connected. So this purine, which is a nitrogenous base, this nitrogenous base is connected to a pentose sugar, and this is connected via a bond, this bond, and this is known as beta N glycosidic bond. Okay, so this structure that here is shown, this is your nucleoside structure. So this is a nucleoside, and since the base here in the example that I've taken is adenine, so this is a adenosine. Okay, I hope it's clear. Let's take another example. So here, as you can see, this is what? This is a cytosine. Okay, now this cytosine is again connected to a pentose sugar, as you can see here, to the carbon 1 of the pentose sugar. What is connected? The nitrogen 1 of cytosine. So, and cytosine is a pyrimidine. So in pyrimidines, they are connected to your pentose sugar by nitrogen 1. Whereas your purines, they are connected to your pentose sugar by nitrogen 9. Okay, remember that. And again, the bond is your N beta glycosidic bond. So the nitrogen 1 of pyrimidines joins with carbon 1 of your pentose sugar by beta N glycosidic bond. And in purines, the nitrogen 9 of purines, it joins with carbon 1 of pentose sugar via beta N glycosidic bond. And what is formed? Your adenosines are nucleosides, basically, which can be adenosine, guanosine, and etc., cytosine and all. So they are formed. Okay, so this is your pentose sugar plus a nitrogenous base, and you end up with nucleoside. Okay, now talking about nucleotides. Now to this nucleoside, if you tie up a phosphate, okay, it becomes a nucleotide. So that is how you can remember. That is how is a nucleotide formed. When to a nucleoside you tie a phosphate. Remember that you're tying a phosphate to a nucleoside, you end up with a nucleotide. Okay, and where are you tying this? At the carbon 5 of the pentose sugar, a phosphate group is attached via ester bond to form nucleotide. So at carbon 5 of pentose sugar via ester bond, you attach a phosphate, okay, and you end up with a nucleotide. Okay, I hope it's clear. So nucleotides, they can have one, two, or three phosphates. If they are having one phosphate at carbon 5, then it is a monophosphate. If they're having two phosphate, it is a diphosphate. And if they're having three phosphate, it is a triphosphate. Okay. So the further phosphates that are attached, they are attached by anhydride bonds. Okay. If I show you the diagram, it will become more clear. Here, have a look. This was your nitrogenous base. Okay, this is your nitrogenous base, and this is your pentose sugar. So your pentose sugar via carbon 1, and this is beta N glycosidic bond. This connected to nitrogen 9. This is a purine, okay? And this is forming a nucleoside. Now this nucleoside, as you can see, it is tied up to a phosphate group here. Okay. Now, how is it tied? This is the carbon 5 in the pentose sugar. So the carbon 5 of pentose is attached to a phosphate. Okay, and since this is just one phosphate, this is the monophosphate. Okay, now you can have more phosphates. See, this is one phosphate and two phosphate. If only two phosphates are there, it will become a diphosphate. But here in this picture, there are three phosphates group. So this is a triphosphate. Okay, so this is a nitrogenous base. This is a pentose sugar, and pentose sugar plus nitrogenous base, they are forming a nucleoside. And when this nucleoside is tied to a phosphate, it becomes a nucleotide. Now you have to join these nucleotides together to form a nucleic acid. Okay. So now let's see how a dinucleotide is formed. This is how you find forming a dinucleotide via phosphodiester linkage. So see, this is here. Now simply I've just written base. Okay, the entire structure, the different bases, everything has been defined. So this is your base. Okay, your nitrogenous base attached to the carbon 1 of pentose sugar. Okay, now to the carbon 5, what are you having? You're having a phosphate group attached. The carbon 5 is outside the ring. Okay, this is carbon 5. Okay, so a phosphate group is attached. So this is your nucleotide 1. Okay, now how is a phosphodiester or dinucleotide formed? What is a dinucleotide? That is, you are joining two nucleotides together. Okay, so this is the structure of nucleotide 1. Now, how is the bond formed? The 3' hydroxyl, okay, of the first nucleotide is engaged in a bond with the phosphate which was attached to the 5' of the second nucleotide. This is the second nucleotide, nucleotide 2, a base, a pentose sugar, and to this pentose sugar you are having at the carbon 5, a phosphate was attached. Okay, so now this phosphate of the second nucleotide is forming a bond with the hydroxyl, 3' hydroxyl of the first nucleotide. Okay. And what is this bond form? This is a phosphodiester linkage, 3' to 5' phosphodiester bond. Okay. So how is a nucleotide formed? Nucleotide is formed by formation of bond between the 3' hydroxyl group of nucleotide 1 and the 5' phosphate, as you can see here, 5' phosphate of nucleotide 2. Okay, and a phosphodiester bond is formed. So this is your dinucleotide. And if we talk about the polarity, so we can say now similarly your more nucleotides will be added to this 3' or hydroxyl and all. So you can see only the first and the last nucleotides they are having free functional group. Okay, so the first nucleotide it is having a free phosphate at the fifth position, and the last nucleotide it will have a free hydroxyl at the third position, and the intermediate nucleotide, that is the middle nucleotides, they will have no free functional groups. Okay, so with this we come to the end of the structure of nucleotides. I hope it was clear. Now let's move on to the metabolism. So we'll begin with purine metabolism first. Okay. Purine metabolism, like I just said, all girls are pure. So if you remember, Pu is purine and all girls, that means your adenine and guanine. So we're going to talk about the synthesis of adenine and guanine first, that is purine synthesis. Okay. Now, this purine synthesis, there are two pathways for it. That is de novo pathway, that is one way to synthesize, and the other is salvage pathway. Okay. Although in diet you have a lot of purine, but that purine that cannot be incorporated in the DNA. So the body has to synthesize its own purine. Okay. Now the site for the synthesis, all organs, especially liver. And what is the organelle? It is your cytoplasm. Okay. And where does the purine synthesis does not take place, or do not take place in your erythrocytes, your leukocytes, your brain and bone marrow. So these organs, these tissues, they are solely dependent on salvage pathway. Okay. So I'll repeat myself again. We're talking about purine metabolism right now. That is synthesis of purine, followed by we'll talk about its catabolism. That is breakdown of purines and the clinical disorders associated with it. So we're going to talk about in purine, adenine and guanine, because those are the most common purine bases. Okay, and where is this purine synthesis taking place? It is taking place especially in the liver, in the cytoplasm, and it does not take place in the erythrocytes, leukocytes, brain and bone marrow. So these organs, the exceptions, these organs, they are solely dependent on salvage pathway for purine synthesis. Okay, now before starting with purine synthesis, let me talk about the sources of intermediate purine ring. Okay, so here if you have a look at this diagram here, so what can you see? This is a purine ring. Okay, you can see there's nitrogen at first position, at third position, at seventh position, and ninth position. Okay, so this is you can see two rings, nine membered, heterogeneous with carbon and nitrogen. So this is a purine ring. Now there are different sources that are contributing to this purine ring. So let's take them one by one. Firstly, your nitrogen 1, the amino group of aspartate. So the aspartate is giving its amino group to form the nitrogen 1 of this purine ring. The carbon 2 of this purine ring, it is coming from formal tetrahydrofolic acid. Then Your nitrogen 3 and nitrogen 9, they are coming from the amide group of glutamine. Then your carbon 8 is coming from methylene tetrahydrofolic acid. Then your carbon 4, 5 and nitrogen 7, as you can see here, they are coming from glycine. Okay, so your glycine, it only has two carbon and one nitrogen. We all know the structure of glycine. It only has two carbon and one nitrogen. Okay, so two carbon and one nitrogen, you can see they have been contributed. So almost the entire of glycine is incorporated in the purine ring. Then your carbon 6 of the purine ring, it is coming from respiratory carbon dioxide. Okay, so these are the sources of your purine ring. Okay, where are the different carbons, the different nitrogen coming from? So now you see the importance of folic acid in DNA synthesis. Formal tetrahydrofolic acid is contributing the carbon 2 of your purine ring, and methylene tetrahydrofolic acid is contributing carbon 8. So in case of folic acid deficiency, what will happen? There will be decreased DNA synthesis causing megaloblastic anemia. So that is why your folic acid is very important in DNA synthesis. Now let's move on to de novo synthesis. Okay, moving on to de novo synthesis of purine bases. Okay, so what do you need to synthesize? You are supposed to synthesize a purine nucleotide. So you will need to have a in a nucleotide, you will need to have a nitrogenous base, you will need to have a sugar, and you will need to have a phosphate. That is how your nucleotide is being synthesized. Okay, a purine nucleotide. So you will have a purine base, a phosphate, and a sugar. So you're starting with, this is the first reaction of purine synthesis, de novo synthesis. Okay, so you're starting with ribose 5 phosphate, as you can see here, ribose 5-phosphate. So what is ribose? Ribose is a sugar and phosphate. So you already have sugar and phosphate. Okay. Now on this sugar and phosphate, different components of the purine ring are going to be added. Okay. So you're already beginning with the sugar and phosphate. You can see this is the ribose sugar. And to this ribose sugar, a phosphate is attached. Now what happens? The enzyme acting is PRPP synthetase. And this PRPP synthetase is going to incorporate a pyrophosphate at position 1 to form 5-ribosyl 1-pyrophosphate, that is PRPP. Okay, and this pyrophosphate, it is being donated by your ATP. Okay, once again, I will repeat myself. So in de novo synthesis, your starting material substrate is ribose 5-phosphate. So ribose 5-phosphate, you already have a pentose sugar, that is ribose, and to it a phosphate is attached. So you have a phosphate, you have a ribose. Now on this phosphate and ribose, you are supposed to add different components of the purine ring, and you will end up with a purine nucleotide. So the first reaction is the reaction that is carried out by PRPP synthetase. So this PRPP synthetase, it is going to incorporate a pyrophosphate, and this pyrophosphate is donated by ATP at position 1. So you end up with 5-phosphoribosyl pyrophosphate, that is PRPP. So in the first step, you are synthesizing PRPP. And what is PRPP? It is 5-phosphoribosyl pyrophosphate. So in short, we are everywhere going to use PRPP. So don't forget what is PRPP. Okay. And the activator of this step is your inorganic phosphate, and inhibitors are your purine ribonucleotides. Okay, and also this step is the preliminary step. This is a regulatory step, but this is not a rate limiting step because this is not a committed step. The PRPP that is formed here, it can go into other fates as well. Okay, it can go for pyrimidine synthesis, NAD synthesis, salvage pathway. Okay, so this PRPP, it is not committed only for purine synthesis. Therefore, this is a regulatory step, but since it is not a committed step, so this is not the rate limiting step of this cycle, because this PRPP has other fates also, like pyrimidine synthesis, NAD plus synthesis, salvage pathway, etc. Okay. Now moving on to the next steps of the cycle. So you have PRPP, that is 5-phosphoribosyl pyrophosphate. Now to this PRPP, what is going to happen? An enzyme known as glutamine phosphoribosyl pyrophosphate aminotransferase is going to act, and your glutamine is going to donate nitrogen 9. Okay, this is nitrogen 9. To PRPP, nitrogen 9 is added first. Okay, and who is the contributor? Your glutamine is the contributor. Okay, so this PRPP that was formed in the first step, nitrogen 9 is added. The enzyme I've already mentioned, and the contributor of this nitrogen is your glutamine. Why, how I'm so sure that nitrogen 9 is being added? Because in purine ring you have so many nitrogens. Okay, four nitrogens are there. But we have just discussed the structure that when you're forming a nucleoside, that is when a pentose sugar is joining with a nitrogenous base. And if it is a purine base, then remember that I said that nitrogen 9 binds to the carbon 1 of your pentose sugar in a purine. In pyrimidine, nitrogen 1 of the pyrimidine was forming a glycosidic bond with the carbon 1 of the pentose sugar. But in purines, the nitrogen 9 of purine was forming a beta and glycosidic bond with the carbon 1 of the pentose sugar. Okay. So now to this pentose sugar, you're adding a nitrogen. So obviously that nitrogen has to be nitrogen 9. Okay. So your nitrogen 9 has been added, and the contributor for this nitrogen is your glutamine. So ribose sugar is attached to purine by nitrogen 9 of purine. So we know that this nitrogen that has been contributed by glutamine, this is the nitrogen 9. Now you end up with what? 5-phosphoribosyl amine. Okay, so you have this ribose sugar, the phosphate was already there, and now you are having an amino group at the first position. Okay, now this 5-phosphoribosyl amine is going to undergo various steps. Okay, a number of steps are going to take place, and in each of these steps a source of purine ring is going to be added. Okay, like first what is going to come? Glycine is going to come, then your tetrahydrofolic acid is going to come, then glutamine is going to come. Each one of them are going to contribute their carbons and their nitrogen, whatever they're supposed to contribute. Okay, so through various steps, as you can see, various intermediates being formed, gar, fagam, air, all these, you finally end up with, in the end, IMP, that is inosine monophosphate. Okay, and this inosine monophosphate, it is the first purine nucleotide that is synthesized. So this is the first purine nucleotide that is synthesized. Now from this IMP, you can get AMP and GMP. So if the amino group is donated by aspartate, amino group donated by aspartate, you end up with AMP, that is adenosine monophosphate. And if the amino group is donated by glutamine, you end up with GMP. Okay, so I'll repeat this once again. That is de novo synthesis for you. So in de novo synthesis, you're beginning with ribose 5-phosphate. On this ribose 5-phosphate, you are adding at the first position a pyrophosphate by PRPP synthetase. Okay, and you end up with PRPP. Now this PRPP, to this PRPP, the nitrogen 9 is going to be added. The contributor is glutamine. This is the rate limiting step. Okay, rate limiting and also the committed step. Now you end up with 5-phosphoribosylamine. Okay, so to the ribose 5-phosphate, add the carbon 1, you have the nitrogen 9 of the purine ring attached that has been contributed by glutamine. Now what is going to happen? Different contributors of the purine ring, different sources are going to come. Like you can see glycine is going to give its 2 carbon and nitrogen. Then you're going to have tetrahydrofolic acid, it is going to give its carbon. Then you have glutamine, it is going to give its nitrogen. Okay, so all the contributors are going to come, and ultimately you can see the ring being formed. This was the carbon 9, then this is the contribution of glycine taking place. Then you can see the contribution of tetrahydrofolic acid taking place. Then you can see the contribution of glutamate taking place. Ultimately, you can see the ring coming into shape, and finally you end up with inosine monophosphate. Okay, so this is the first purine that is synthesized. Now to this IMP, if amino group is donated by aspartate, you end up with AMP. If the amino group is donated by glutamine, you end up with GMP. So that was about de novo synthesis of the purines. Now, like I told you, that the synthesis of purines, it has two pathways. One was de novo, okay? So the pathways for biosynthesis of purine nucleotides. One is your de novo, that is de novo means a new. That is nucleotide bases, they are synthesized from simpler compounds. And the second is salvage pathway. Now, what is the salvage pathway? Here, the preformed bases, they are recovered and connected to a ribose unit. Okay. So what is happening in salvage pathway? This ensures recycling of purines formed by degradation of nucleotides. So your nucleotides, they are degrading, and then the recycling of purines is going to take place. Okay, so you're basically salvaging those purine bases. Okay, so you can understand like this. This is your cell. Okay, cell lysis is going to take place. So when cell lysis occurs, what happens? Your DNA comes out. Cell is lysed, your DNA comes out. Now on this DNA, what is being added? DNA is going to act. Okay. So on this DNA is going to act, and your nucleotides are liberated. Now on these nucleotides, what is going to act? Nucleotidases is going to act. And it is going to remove pentose sugar and your phosphate. Okay. So your pentose sugar and phosphate is released. So when sugar is removed, phosphate is removed, what do you end up with? You end up with bases. Okay, you end up with purine bases. Now these purine bases, they can catabolize to form uric acid. I'll repeat myself again. Cell is lysed. Okay, on lysis of the cell, what happens? DNA comes out. Okay, now this DNA is acted upon by DNAs. Okay, so it is going to cut that DNA into its monomers, that is nucleotides. So your nucleotides are liberated. Now you have the monomers. Now on these nucleotides, nucleotidase is going to act, and that is going to remove the sugar and the phosphate. So sugar and phosphate are removed. Now you are ending up with purine bases. Okay, now these purine bases, they can catabolize, get degraded into uric acid. But the body will not do so, because the body has spent a lot of energy in synthesizing these purine bases. Okay, so the body will not want this energy to go into waste. So the body tries to save these purine bases. Okay, so these purine bases, they are going to be salvaged. Okay, and then salvaged back to purine nucleotides. These purine bases, they're going to be salvaged back to purine nucleotides. That is all the sugar, pentose sugar and the phosphate that has been removed. It's going to be added back to these purine bases, and it's going to be salvaged back to purine nucleotides. And then this purine nucleotide can again be used for DNA synthesis. So in this way, body is trying to conserve energy. Okay. So now you understand that is what is the other way. One was the de novo synthesis, that you were forming the purine bases anew, the purine nucleotides anew from simpler compounds. Okay. That is how you were synthesizing. And here what is happening? You are trying to recycle the purine bases that you're getting from nucleotide degradation. So from the DNA, you are getting the nucleotides. From the nucleotides, the nucleotidase is going to act on those nucleotides. The pentose sugar and phosphate is removed, and you end up with these purine bases. But in order to conserve energy, the body is going to salvage these purine bases, and then they are going to be salvaged into purine nucleotides, which can again be used for DNA synthesis. Okay. Now, in salvage pathway, there are two types of reactions taking place. Okay, again, now salvage pathway 1 and salvage pathway 2. So, in salvage pathway, where you are salvaging the purine bases, that is, the purine base is being converted to a purine nucleotide, that is taking through a phosphoribosylation reaction. Okay, and where you are converting salvaging a purine nucleoside. Okay, in a nucleoside, you're only lacking a phosphate. So when you are salvaging a purine nucleoside and that purine nucleoside is being converted into purine nucleotide, that is through a phosphorylation reaction. Okay, I'll write this down for you so it becomes more clear. So you have purine nucleotides. Okay. Now these purine nucleotides, since they are nucleotide, they have the purine base, nitrogenous base. They also have the pentose sugar. They also have the phosphates. Okay. Now what happens? From the nucleotides, if you remove the phosphate, you end up with purine nucleosides. Okay. Now, from the nucleosides, if you again remove the pentose sugar, because what were nucleosides? They were pentose sugars attached with the nitrogenous bases. Now, your sugar has also been removed, so you're only left with purine base. Simple up till now. Okay. Now in salvage pathway, I'm saying that there are two types of reactions taking place. If you're salvaging purine bases, that is this purine base, if you're salvaging this back to purine nucleotide, then what all do you need to add? You need to add pentose sugar and phosphate. Okay, so you need to add phosphate plus pentose sugar to salvage this purine base back to purine nucleotide. So the donor for this is PRPP, phosphoribosal pyrophosphate. Because remember the structure of phosphoribosal pyrophosphate, it has a ribose and it has a phosphate group at the first position, and then a pyrophosphate was added at first position. Okay, so the donor of this pentose sugar that you need and the phosphate that you need to add to this purine base to salvage it back to a purine nucleotide, the donor is PRPP. Okay, and this is known as a phosphoribosylation reaction. Phosphoribosylation. Why is it known as phosphoribosylation? Because you're adding a phosphate as well as a ribose. Okay, so known as phosphoribosylation, and this is salvage 1. Now, if you want to salvage a purine nucleoside, okay, so nucleoside is just not having a phosphate group. It is having the purine base. It is having the pentose sugar. You only need to add a phosphate group. And who is the donor of phosphate group? ATP. Okay. So you're just adding a phosphate groups and ATP is required. And this ATP is changed into ADP and you're adding a phosphate. So this is known as phosphorylation. And this is brought about by the enzyme kinase. Okay. And this is salvage 2. Okay. Now let's talk about the reaction of salvage 1. So for adenine, if your purine base is adenine, your donor for the phosphate and the ribose sugar is your PRPP, like I told you. Okay, so your phosphate and pentose sugar being donated by PRPP, and the enzyme is adenine phosphoribosal transferase, that is represented as APRTase. Adenine phosphoribosal transferase, because your phosphate and ribose is being transferred, okay, and a pyrophosphate is being released, and to this adenine a phosphate and a ribose is being transferred, you end up with AMP. Similarly, for guanine and hypoxanthine, the enzyme is same. That is HGPRTase, that is hypoxanthine guanine phosphoribosal transferase. Okay, so to guanine, if you add a phosphate and a ribose sugar, you end up with GMP and a pyrophosphate is released. Similarly to hypoxanthine, you end up with IMP, that is inosine monophosphate. So the nitrogenous base in inosine is your hypoxanthine. Okay. So remember, in inosine, nitrogenous base in inosine is hypoxanthine. Okay. And in phosphorylation, what is happening to the adenosine? Okay, so like you have a purine nucleoside. Okay, so you only have to add a phosphate, so that will be taking place through a kinase. So to the adenosine or to the guanosine via the kinase enzyme, and ATP is required, you end up with AMP and GMP. Okay, so that was about the salvage pathway reactions. Now let's talk about the significance of salvage pathway. So what is the significance? One, I've already told you that it saves energy because the body had spent a lot of energy in synthesizing your purine bases. Okay, so the body does not want it go into waste. So one of the significance is that it saves energy. The second being there is effective recycling. Then it is important in organs where de novo synthesis is not operating, especially in your RBCs and the brain. So in organs where your de novo synthesis is not taking place, their salvage pathway becomes very important. Then HGPRT's highest concentration is present in basal ganglia. Okay. So HGPRT's highest concentration is in basal ganglion of the brain. Okay. So what happens? Therefore, in case of deficiency of HGPRTs, the neurological defects will be manifested. Okay, because your brain is completely dependent on salvage pathway. Now, there is a disorder associated with deficiency of HGPRTase enzyme. Okay, so because of this deficiency of HGPRTase, there is a syndrome or a disorder occurring which is known as Lesch-Nyhan syndrome. This is important, frequently asked in short notes. So this is an X-linked recessive disorder, X-linked recessive. So that is it be mostly occurring in males because since it is X-linked, so your males, they have only a single copy of X chromosome. So if they have a mutant copy, it will be manifested. In females, this is rarely occurring because since it is X-linked recessive, so the females, they will need to have both the defective copies of X, and females are having two copies of X, so there's a rare chance that both the copies are defective. Okay, so it is mainly manifested in males. And what is happening in Lesch-Nyhan? There is complete deficiency of HGPRTs. Complete deficiency of HGPRTase enzyme. So what was your HGPRTase doing? It was salvaging your hypoxanthine and guanine. Okay. So your hypoxanthine or guanine, when PRPP was donated, phosphate and ribose sugar, this enzyme was acting HGPRTase, and what was being formed? IMP or GMP respectively was being formed. Now what happens? Because of the deficiency of this enzyme, there is no HGPRTase, there's complete deficiency. What will happen? Your hypoxanthine and guanine, they will not be salvaged anymore, so they will increase. Also, your starting material PRP will also decrease. And what will will also increase. And what will decrease? Your IMP and GMP, they will not be synthesized, so that will decrease. So in case of deficiency, there is an inability to salvage your hypoxanthine and guanine. Okay, as a result of which you will have excessive amounts of uric acid, because when your hypoxanthine and guanine, they are increasing, obviously they will be degraded. Okay, and the degradation product of your purine is uric acid. When your purines, they are catabolized, they form, the end product is uric acid. So what will happen? Your uric acid will increase. Okay, and also, like I told you, your PRPP was increasing. So your levels of PRPP are also increasing because HGPRTase is not there. So hypoxanthine guanine is increasing. The other substrate PRPP, your PRPP is also decreasing. Your IMP and GMP, they are decreasing. Now, if you remember the rate limiting enzyme in purine synthesis, the second step that I told you was the rate limiting committed step, which was contributing the nitrogen 9 of the purine. That was regulated by your excessive substrates and the inhibitors. I'll show you once again here. I'm talking about this enzyme, phosphoribosyl pyrophosphate amidotransferase. Okay, so the inhibitors for this are your AMP and GMP. So your AMP and GMP, they have decreased, and your activated PRPP has increased. So your activator has increased, your inhibitor has decreased. So this enzyme will work more. So there will be increased purine synthesis. Okay, are you getting my point? Because this enzyme is going to be activated. So there is increased purine synthesis, but there is decreased purine reutilization because your salvage pathway is not taking place, as a result of which there will be more amounts of purine being synthesized. More purines being synthesized means more degradation, and hence there will be more uric acid formation taking place, thus leading to hyperuricemia. So in Lesch-Nyhan syndrome, your hyperuricemia is going to be manifested. Then there is also intellectual disability. There's neurological manifestations, as we know that in the brain, your salvage pathway was the only source of purine synthesis. Okay, now that's going to be affected. So you'll have neurological manifestations. And then there is another very important clinical manifestation, that is compulsive self-mutilation. Okay, so the person is going to harm themselves, biting lips, finger, toes, etc. So these are the clinical disorders associated with Lesch-Nyhan. I hope it's clear. Now let's talk about the treatment of Lesch-Nyhan syndrome. So talking about the treatment, there is only symptomatic treatment present. Allopurinol can be given. Allopurinol, since allopurinol, it is an inhibitor of xanthine oxidase. Okay, so it inhibits xanthine oxidase. And xanthine oxidase, when we'll talk about degradation of purine nucleotides, you'll see this is an enzyme which is forming uric acid. Okay, so your uric acid formation will be decreased. Okay, so production of uric acid is going to be decreased if you give allopurinol, since it is inhibiting xanthine oxidase. Then another treatment can be high fluid intakes. Along with alkali. Why? Because there is decreased crystallization of uric acid in alkaline mediums. Okay, so the uric acid crystals are going to get dissolved in alkaline medium. Therefore, a high fluid intake with alkali is suggested as a form of treatment. Okay, so your hyperuricemia results in the formation of uric acid stones in the kidneys and deposition of urate crystals in the joints and other soft tissues. So for that, to dissolve the uric acid crystals, alkali intake is recommended. Now, if there is partial deficiency of this HGPRTase, the disease that occurs is known as Kelly-Sigmiller syndrome. So Kelly-Sigmiller syndrome is a milder version of HGPRTase deficiency. There's partial deficiency of HG PRTs, and in Lesch-Nyhan there was complete deficiency of HGPRTs. Okay, So with this, we come to the end of today's class. A small topic that is purine nucleotide degradation is left. So when I take the next class, I'll quickly cover this before beginning with enzymes tomorrow. Okay, so for tomorrow's class, we have enzymes. If you have any queries, you can ask. Thank you for watching. See you in the next class tomorrow.